Well, we can go ahead and get started then. Thanks everyone for joining us for our May meeting. We're going to be talking cryptosporidium today. I see Brianna Loke has joined the conversation. Brianna, are you ready to speak?
Hey Ben, I am here.
Oh,
Were you guys going to share some
oh,
slides first and then I'll
yeah, I
follow up?
can totally, for sure.
Okay, thanks.
Sorry about that. Let me get ready then. Okay, everybody can see that.
Yes.
All right, great. Okay, so we kind of wanted this month to, we were doing kind of a little series where we would talk about the Department of Health and Human Services and the NPHL's roles in some of this testing and surveillance that goes on. We kind of got away from that for a little while and wanted to get back into it. So we asked Brianna from the Department of Health and Human Services to talk about cryptosporidium, and I'm going to talk a little bit about what it is we do here in the Nebraska Public Health Lab. Um, I believe, yeah. So we've got Amy and Stevie and Emily are here to answer any questions or correct any of my mistakes. So we will go on here. So cryptosporidium, this is a modified acid fast stain for cryptosporidium. This is stained directly from stool. Stool tests that are positive for cryptosporidium, we're asking, be forwarded to the NPHL for additional PCR and sequencing if appropriate. And so the stool samples at cryptosporidium is a target on the GIP panel. So whether it's detected, through microscopy or PCR, we are asking that those samples be forwarded. There is no pre-approval needed from the local health department, and we ask that you order NPHL BK. We perform that testing Monday through Friday, turnaround time up to 7 days. I want to note here that sequencing, if sequencing is performed, that is for epi purposes only, and those sequencing results do not get reported back to clinical providers. And specimens, we can accept fresh stool, stool in protofix, stools in 10% formalin. We ask for at least 3 mils, and stool samples are generally stable in ambient air. If you are sending in a fresh stool sample, you want to send that in a little sooner than later. I have to... Okay, so for fecal samples that test positive for cryptosporidium, normally it's contained in cryptosporidium oocysts. The DNA is extracted using this fast DNA spin kit for soil. The process begins by mechanically breaking open and lysing the oocysts using a homogenizer, and then lysed cells are subjected to a series of spin and wash steps that lead to the isolation of the DNA. So to start, around 200 microliters of fecal sample is added to a labeled, no label here, a labeled lysing matrix tube, which is of course done in the biosafety cabinet. Samples that arrive in potassium dichromate, those get a couple extra washing steps. But to this tube here, a couple of solution buffers will be added, and there's a lysing agent inside, and those beads, obviously, and they're loaded onto a mechanical homogenizer, which looks like this. And this is a bead beater that's designed to homogenize, grind, and lyse biological samples. It shakes in a manner that increases collision between the sample and the specimen lysing matrix. And then following homogenization, the tubes go back into a centrifugation step. There's centrifuge to separate the supernatant, which will contain the DNA that we're trying to isolate. This is transferred to a clean tube, and a precipitate solution is added to the tube to remove any remaining protein or possible contaminants. And it's once again mixed and centrifuged, and supernatant once again transferred to a clean tube. And to that, a binding matrix is added, a suspension of silica particles. When in the presence of high salt content, this matrix will bind to the DNA. And once the DNA is bound to the matrix, the solution is transferred to a filter tube to separate it from the rest of the solution. Looks like this. The DNA bound to the silica matrix is trapped on the filter membrane, and contaminants that remain in the solution pass through with the liquid to the bottom there. Additional impurities are washed, and then at the end, a low salt buffer is added, which separates the DNA from that binding matrix, which leaves you with your high quality purified DNA. So following extraction, a master mix, the primers, and the freshly extracted cryptosporidium DNA are added to reaction tubes and loaded onto a thermocycler, which undergoes a series of temperature changes to activate and deactivate different enzymes in the master mix, which promotes the DNA replication. Along with extraction steps is an extraction control to make sure the samples weren't contaminated, and alongside the amplification is positive and negative PCR controls to make sure that the process works properly. Following the initial PCR, a second nested PCR is performed using the products from the first. I'll kind of cover this a little bit more in a second here. So the first PCR is performed. The products from that PCR are used along with new primers to perform a nested PCR to increase the specificity and improve amplification of the DNA that we're looking for. And then following that nested PCR, if those products are detected on gel electrophoresis, or those products will be detected with gel electrophoresis, which looks like this. So following that nested PCR, products get loaded into the electrophoresis cassette, and if any sample shows an appropriately sized amplification band, that'll get purified further for sequencing. If no bands appear on the first round of nested PCR, it'll get reflexed to a second round looking for a different gene. I'll go over that in just a second. Which then, similar to this first round, will get gel electrophoresis, and any bands will be forwarded on for sequencing. And after the second round of electrophoresis, any samples with no bands are reported as negative. And so here's kind of what the algorithm for testing looks like. The first round of PCR and nested PCR, we're looking for the GP60 gene isolation, and from that we can determine Cryptosporidium parvum or Cryptosporidium hominis. If the gel electrophoresis is negative for the GP60, it gets reflexed to the 18S, which goes through an original round and then a nested round of PCR to isolate it, to isolate any of that DNA. And then the sequencing, if any of those are positive, they'll be forwarded for sequencing. So those products of 18S or GP60 DNA are forwarded for standard Sanger sequencing. The raw data generated is trimmed and cleaned using a special software and then compared to a known Cryptosporidium reference sequence. From the GP60 amplification, Cryptosporidium hominis and Cryptosporidium parvum subtype family and subtypes can be determined. And then from the 18S products, we can identify just Cryptosporidium species. This screenshot, which was provided by Amy Roden, was taken from bidirectional Sanger sequencing. It's an analysis of a crypto sample. And what we want to point out here is the tandem repeat section. Get my pointer here. Oops. This tandem repeat section in the DNA, because that helps us determine the subtype family and the subtype. So, and again, this comes from the species, the Cryptosporidium parvum or the hominis, and that's only available if the Cryptosporidium is detected via the GP60 locus. And they are named. You have the species, they have a subtype family, and then a subtype. For Cryptosporidium hominis, the subtype family would be listed as 1A, 1B, 1D. For Cryptosporidium parvum, like we see here, you get your 2A, 2B, 2C, and this is a very broad overview of the subtyping and subtype family, which is going to be followed by the number of either TCA, TCG, or TCT repeats, which would be presented by the letter A, G, or T. So we have our letter A there, followed by the G. And in the Cryptosporidium parvum 2A subtype family, like we see, a few subtypes have two copies of a ACA-TCA sequence right after the trinucleotide repeats, which is represented by this R1. So we use these subtype families and subtypes. Brianna will use these in her surveillance and outbreak investigations. So that is a short, brief overview of the Cryptosporidium testing we do here. Are there any questions for the lab before we hand it over to Brianna? I know I spoke kind of quickly throughout that whole thing. Okay. Brianna, are you
I'm still here.
ready to go?
Yes, and sorry I don't have any slides, but
Okay.
thanks for the opportunity. So I will just kind of touch on the epidemiologic side of what we do for Cryptosporidium. So first, a little background. Cryptosporidium is a reportable disease, and we at Public Health are notified as soon as we receive that positive lab report in our surveillance system. The protocol is local health departments will create a case investigation and they'll attempt to contact the patient to ask about risk factors and exposures they did before they became sick. And then all this information is collected and documented in a data collection tool that we use. It's called REDCap. We collect enhanced Cryptosporidium risk factors as a part of the CryptoNet project that we are funded funded through CDC. That's another story. Anyways, and we report all this information and data back to the CDC. At the state, we will monitor the data on a statewide level, usually about a weekly basis to look for commonalities across cases. If a cluster is identified, we will work with the local health department to collect additional information or possibly re-interview cases, contact the clinical labs to ensure they send the positive specimens to MPHL, and then we'll notify MPHL about those possible outbreak specimens that they're headed their way. And to share a recent example of how the genotyping was super helpful and supported an outbreak investigation was last fall. We had a crypto outbreak in a childcare facility. The state enteric team, we noticed 7 crypto cases were reported for a central local health department jurisdiction in the month of September, when we knew this was super unusual because really they only see maybe 1 case per month, or average 10 cases per year. So to have 7 crypto cases within a month was pretty alarming. We made sure those specimens were sent to NPHL for genotyping, and we notified them as soon as possible saying, hey, we think this might be part of an outbreak. Can we pretty please have genotyping on these? While interviews were still ongoing, in some cases we couldn't follow up with NPHL. They provided a really quick turnaround with the genotyping. So we got our preliminary results, which really helped us figure out which cases to include or exclude from the outbreak, depending on the genotyping and the species or the subtype. So after narrowing it down, we were able to just focus on the related cases only. And once we had all of our final epi data, after recontacting some cases about childcare exposure, maybe they didn't report it in their initial interview, we were able to find that common link, which was a single childcare facility. What was interesting about this outbreak was that most of the positive cases were actually adults. So it wasn't, when we had some of the preliminary data, it wasn't showing to be a child care facility outbreak because, well, adults aren't at child care facilities. Only 2 kids tested positive while the rest were adults. So that the genotyping, having that information ahead of time really provided some clarity and some answers in the investigation. So we knew who was related. And then once we had that final epi data, and we were able to re-interview cases, we were able to find that link. That some of the adults had children in that childcare. So I haven't seen that type of outbreak before where most of the adults were sick, but only very few kids were tested. Um, so, and then kind of just like a summary of crypto in Nebraska. We don't have very many crypto outbreaks. We may investigate 1 or 2 per year. In terms of surveillance in Nebraska, we've been really on a downward trend with crypto. Probably about 10 years ago, back in 2015, 2016, we were in the upper 200s for crypto cases reported. But now in the last couple of years, we've gone down to the lower 100s. And even last year was at 85, which is the lowest we've seen in the last 10 years. So that's good news. But yeah, so that is kind of the crypto epi summary, an example how having this genotyping capabilities is super helpful for us in the epi world. And I'd be happy to take any questions.
Brianna, just a curiosity, what do you think is the main driver of the downward trend?
That's a great question. I am not sure. Well, actually, back in 2015, and maybe Brian, if you're on, I know there was a large, wasn't there like a water park outbreak back then that could have contributed to some of the upper 200 numbers? I'm not sure. So in 2015 and 16, we were in the upper 200s, so like 275-ish. And then over the, 2017, 18, 19, we were down in, well, here, let me show you. I could show you my chart if that would
Yeah,
help too.
if you have a visual, that would be really helpful. I was just
Okay, hang
trying
on.
to see if I could remember any other outbreaks.
I can answer, guys. We used to have a large number of facilities across the state of Nebraska that were using antigen tests for crypto, and those antigen tests frequently did not confirm out. We, in the last decade, have seen a larger shift towards more molecular-based identifications in that realm, and therefore have seen more actual true infections being identified as opposed to a false positive antigen.
Can you see my screen?
Yes, we can.
Yeah, okay.
Yep.
It's not great, but this is what I have. So yeah, kind of just a consistent downward trend over the years. And in terms of outbreaks, if I think that water one back in 2015, that was a pretty big one. But some of the outbreaks over the last couple of years might just be small, little ones that only involve maybe a handful of cases, at least for confirmed ones. Sure, there's probable, and I could break this out by confirmed and probable, but
yeah, I know we did a lot of communication out to submitting facilities right in the 2018, 2019, 2020 timeframe about those antigen tests and comparing when we had, a biofire positive result coming in and looking at the sequencing results that NPHL was obtaining versus antigen tests coming in and what the true positivity rate of those as confirmed via molecular methods was. So right around that time is when we really did a lot of that communication and brought to light that those antigen tests were, surprise, not very accurate. So yes, I do think that correlates with that. I do think that we should note, like you said, we don't have a lot of like water parks per se here in Nebraska. especially indoor water parks. But it will be interesting to see how cryptosporidosis cases is reflected in the near future when that large indoor water park goes live in the Bellevue area in the coming years.
Mhm.
This is Pete. We're pointing fingers at water parks, but I agree that's a good place to look. I'd be interested to know, Brianna, parvum and hominis are the most common species detected, I'm assuming.
Mhm.
I guess I'm interested to know what role animals play in transmission of this disease versus humans, because parvum comes from animals, hominis comes from humans.
Mhm.
Have you studied anything along those lines? Because we are a cattle state. Do you see more parvum coming from the western part of the state, for instance?
Yeah, a while ago I did that CDC MMWR about cryptosporidium between rural and urban areas.
Yes.
And yeah, we did see uh, parvum associated with like more rural areas, cattle, farm animals, and then the crypto hominis was more urban and like
Okay.
childcare facilities. And yeah, so when we got the results for that childcare outbreak and it was hominis, I was like, ooh, like this, I have a strong feeling it's a childcare situation.
Okay.
Even though it was like central Nebraska, but it's still pretty populous in that area. So yeah, that was like my first clue because I remember that write up where
Okay. Yeah.
yeah.
So it does, there is a pattern there.
Mm-hmm.
It's
Yeah.
also interesting to me when we see the results, and someday we'll have to put this in a chart, but all the other species that we see of Cryptosporidium, the other animals involved, like chipmunks, and I'm thinking felis cats,
Mm-hmm.
there's quite a variety, I guess is what I'm saying, of different animals that transmit to humans.
Yes, and I know CDC, they in Cedric, where we report the CryptoNet data, they have like all the charts and everything made depending on species from each state. So I'm sure a report could be pulled out from there.
Be pretty cool. I think one of the more common other species was felis, if I remember correctly, some cats.
Yeah, yeah, we do see
Yeah.
that. And then the chipmunk. Didn't chipmunk change though to something
They,
else?
they changed the name.
Yeah, I
Yeah.
can't pronounce the name. I don't know.
No, but I mean that's what this sequencing is enabling us to do, is to look at the variety, because everybody in the past was just Cryptosporidium, period. Now we can actually speciate it, which is showing some interesting data in my mind. So we appreciate everybody sending these samples in, because I think we're learning as we go along what are we seeing when it comes to this particular organism. All good stuff.
All good stuff. Thanks, everyone.
Mhm.
Any additional questions? Got a very knowledgeable crew here. Okay. Well, thank you. Thanks, Brianna, for speaking, and Pete and Emily. We will move on to our section updates then, and we'll start at the top with the HAI AR team.
So for the HAIAR team, we are still working with the C. auris outbreak investigation. This started earlier in the year. This year, we had one last year, but that was completely kind of um, uh, contained, and we have not seen any additional case related to last year's C. auris outbreak, where our belief is that a patient from Arizona, was kind of the one that kind of brought it into the state, and then we have that. So that part is done. We do think that this is a similar scenario, whereas from another state now, a patient brought it into the state, and we saw a cluster of cases associated with that. And I think we have identified about 6 of clade 1 CRS related to that cluster. We still have to do a few more colonization tests and a few more rounds of testing. But it appears to us right now that we might be getting into a containment mode here. We probably in a good containment situation right now with that clade 1 CRS. However, incidentally, we have found two clade 3 CRS cases. At this point, we have no idea how they got introduced to our state. So there have been two identification of clade 3 CRS cases that we don't have an idea how it was kind of introduced in our state. Luckily, we are not seeing further transmission of it right now. But that still so what it tells us is that there are CRS cases in the state that we may not know of. So having said that, why it's all important is that anytime we see a sample that is suspicious, meaning that if we consider that this may not be necessarily it might be possible to misdiagnose based on the system you are using, that CRS can be misidentified as some other organism. Reach out to NPHL, send the sample to them to kind of verify that. Or if there are sterile sterile site culture that are showing Candida, but you cannot go all the way to the species level, reach out to the NPHL on that. We just have to be aware that CRS is in the state, and we have to keep our threshold low to get those specimens tested to identify CRS if it is CRS, so that we can do some containment efforts. We want to delay the spread as much as possible. Eventually, I think it will spread, but the point here is to delay that as much as possible. So that's basically it for the CRS situation. We are also looking at a CPCRE cluster investigation at this moment. Again, that's another area where we don't have that many CPCRE cases in the state, so we can keep that contained also. That will be a great thing. We think we are starting to understand where these cases are coming from, and we are putting some additional investigational effort in that particular area. We'll let you know as we learn more in subsequent meetings on those cluster outbreaks investigation progress. That's about it. Thank you.
Thanks, Dr. Ashraf. Are there any quick questions for Dr. Ashraf concerning CRS or CPCREs? Okay, thanks again. Respiratory illness.
Yeah, thanks, man. This is Derek. I can cover that. So just to start with a quick little update on activity for kind of our main three respiratory viruses that come to mind, influenza, RSV, and SARS-CoV-2. Starting with influenza, activity continues to be pretty low, which is what we would expect for this time of year as summer is approaching. Not really seeing any significant activity with influenza A or influenza B. And still to date this season, we really haven't seen an influenza B wave. I know a lot of other states across the country, and including specifically within the Midwest, have reported the same and not really seeing influenza B. So knock on wood, I don't anticipate seeing any influenza B activity as we're about the summer. Quickly on RSV and SARS-CoV-2, I'm just kind of lump those two together. Both of those viruses activity are also low. I'm not seeing any significant activity there. A couple viruses that we are seeing a little bit of waves and increases in our laboratory data are rhino and enteroviruses, which is not atypical for this time of year. We typically see a little wave of those positives come in, as well as parainfluenza 3. We've seen increases with that virus specifically, and that's just not unique to Nebraska. Some other states across the country have reported similar increases. Again, nothing unusual there. but just wanted to highlight that. Then just a couple routine reminders, as we've mentioned in previous calls. If your lab has any influenza A unsubtypable specimens, please notify us as soon as possible and submit that specimen to NPHL so we can get some subtyping done. And then as summer continues to creep closer and closer, which means we're creeping closer and closer to summer fairs, agricultural fairs, just want to keep in the mind of variant influenza detection. So specifically with influenza A, if you're able to do local subtyping on all your A specimens, that's great. We would like to get as many, if not all, influenza A positives throughout the summer subtype. So if you aren't able to do any subtyping locally, please send those to NPHL. And then, of course, if you happen to learn of a specific exposure locally with a patient who reports an exposure to whether that be swine or birds, because avian flu is still out there, please notify us as well as soon as possible so we can coordinate getting some specimens sent to NPHL for further testing. So with that, happy to take any questions. And if anyone at NPHL wants to add anything, please do too. Thanks.
Thanks, Derek. If there are no questions, we'll move on. I don't know if, Brianna, you have updates for enterics.
Okay, sorry. Thinking. We have started to see the annual salmonella backyard poultry outbreak that happens every year. That happens between like now and September that we'll see a lot of cases. In Nebraska, we average about like 15 to 30 cases a year in that multi-state outbreak. So I guess just a reminder to make sure you're forwarding all your salmonella positives or isolates to NPHL so we can get whole genome sequencing on those to be able to match them to multi-state outbreaks. We have, I believe, one case right now in that outbreak so far. Other than that, let's see. I think that's it.
Okay.
Yep.
Thank you.
Mhm.
Any questions? Okay, we'll move forward to arbovirus.
Yep. Hello, everyone. Right now, the name of the game is ticks. It's tick season. We're right in the prime of it. And just looking, kind of at our syndromic surveillance for visits related to tick bites, we're seeing that increase where we typically see it. We're a little bit below where we were last year at this time. A lot of that's probably due just to the fact that it's been a little bit drier out this year compared to last year around the same time. So there's probably a little bit less tick activity going on compared to last year, and we had really, really good conditions for more ticks. But with that being said, like I said, we are kind of in that prime. We are starting to see some positive tick labs come in. So last year, our highest reported, or typically our highest reported tick-borne disease is ehrlichiosis. Last year, we had a very large outbreak of tularemia. In fact, it was our most cases ever reported, but most of that occurred late after August 1st, which is very unusual. And most of that was related more to what seemed to be like aerosolization and not actual tick bites, where typically we expect most of our tularemia cases to be tick related. So that was something unusual last year. But, we have, Lyme disease, Rocky Mountain spotted fever, ehrlichiosis, tularemia, all those can be locally transmitted. We do have those tick vectors. So that's kind of thing to be on the lookout now. As we go through the season, we'll see likely our tick activity continue to increase until probably right around first part of June, second week of June. That's kind of when we start to see our numbers kind of start to come down in terms of tick bites, visits. And we're, starting our tick surveillance now. Our tick surveillance isn't set up to do, seasonality like our mosquitoes are. It's more for detecting where the tick species and the pathogens they carry are at. So that's a little bit different setup. But outside of that, the only other thing I really wanted to touch upon is, malaria right now. We're actually seeing a number of malaria cases that have come in recently with individuals returning from travel to endemic areas. Last year, we had the most cases of malaria reported since 1947, I believe. So, with this many cases this early on coming in, it just kind of keep it on folks' radar that if there's individuals returning from malaria endemic regions, that report fever and, body aches, headaches, maybe to think, be sure to think about testing for malaria since we're seeing more cases of malaria than we expect for this time of year. But outside of that's all we got right now as we continue the season. We'll eventually get into mosquito season and it'll be West Nile virus season. So that's all I got for now.
Thanks, Jeff. Questions for Jeff, the arbovirus team? Okay, I see Kristen's popped up here, ready to give us an update for TB.
Yes, good afternoon, good morning. I do not have any updates today.
Okay, great. Thank you. And we'll move to the SCD program. Tammy's on. No.
This is Dennis
Oh, hi,
Lishinsky.
Dennis.
I can give a quick update on STDs. So the best news so far is syphilis is down, and almost down maybe about 30% from last year. Last year would have been, I think, our high in quite some time. I haven't gone back to like historical data, but last year we had over 700, like 750 cases. And with that, congenital syphilis is also going down. Last year we had 8 to 10. Right now we're at 2. And for chlamydia and gonorrhea, we're seeing decreases. The decreases with those two have been going on for a while. Gonorrhea since 2020, 2021. Chlamydia about a year later. So those are all trending in the right direction. And for syphilis, it's about time.
Yeah, that is great news. Thank you for the update. Any questions for Dennis? And we'll close out with lead testing.
Hello. So the only thing we have today is that the FDA is alerting that reports of falsely elevated results have occurred when using the ASP Global's RAM Scientific Safety Fill Microcapillary Blood Collection Tubes, that's a mouthful, with the lead care testing systems. These tests may overestimate blood lead levels and give inaccurate results when processing capillary blood samples collected in these Global RAM Scientific SafeTL tubes. The root cause right now is unknown, and the FDA is recommending that these safety tubes not be used with lead care testing systems while this issue is being investigated. And that's all I have for today.
Sandra, is there a resource available where people can get more information on that?
Yes, I can put the FDA
Yeah, that would be great.
link in the chat here.
Great. So are there any questions for Sandra regarding that testing system? And then, okay, so she has, yep, the FDA link in the chat there. Okay, thanks a lot, Sandra. So closing out here, as for NPHL training, we are prepping the Gram Stain Challenge if you are a participant in that. The packaging and shipping invitation for our June class has gone out. So if you have employees needing packaging and shipping training, be sure to get them signed up for that. And yeah, our rule out and refer testing or training is going to be right around the corner now because summer moves quickly, and that's going to be at the end of September, beginning of October. So look for invitations for that as well. So are there any additional comments, any last minute comments or questions? Okay, so our next meeting is not going to be on the second Monday of the month in June. Becky and I are going to be traveling, so we're going to back it up a week. That's a tentative plan for right now. If there's any big issues with that, let us know so we can work around any scheduling issues with the DHHS or the labs. So that's all I have. So laboratorians, thanks for joining. You're free to jump off.